This is Unit 8B, Part 1. During and immediately after the implantation, the blastocyst, which has a single cell layer, undergoes a process called gastrulation to become a multi-layered gastrula. So there's more than one layer, and we're talking about this area in here, right there. So the inner part, the inner cell mast, is the embryoblast, and it divides into two layers during gastrulation or formation of the gastrula. A upper epiblast, and just a second, a upper epiblast and a lower hypoblast. So here you see the epiblast and here you see the hypoblast, and we're talking about these blue colored cells there versus, let me get a better color here, these blue cells here, that layer there, versus the hypoblast, lower hypoblast. So here we have a 8-day blastocyst. By 12 days, the process is being completed, has been completed in terms of the implantation, and the gastrulation process is ongoing, and you have that upper epiblast and lower hypoblast, and together these two layers form the embryonic disc, and that goes on to develop further, and you go towards chorionic development. Chorionic villi are further developed. There's the chorion. And I believe on the next few slides it talks about the layers, and chorion is one of them. Yeah, so the embryonic membranes, or the extraembryonic membranes, these membranes form in the first 2 or 3 weeks of development. So that's what's going on in here. And the membranes that are extraembryonic, outside of the embryo, are the amnion, the yolk sac, the allantois, and the chorion. And here you see them, the chorion, the amnion, the yolk sac, and the allantois here. Okay, and these four are described in the following slides. When they talk about the yolk sac, it's the membrane that surrounds the yolk inside the yolk sac. It's not, it's this whole structure, but the membrane they're talking about is the membrane that surrounds that. So they're talking about this here. They're talking about this here. Okay. All right, let's go through them. Oh, I should add, it doesn't want to move forward. I don't know why. It doesn't want to advance. Hold on. Okay. So the amnion here, described and shown there, is a layer of cells that forms a membrane that'll be filled with amniotic fluid. And you see that amniotic fluid as a light blue material. So, and that's filling that amniotic cavity. And this provides a protective environment that the embryo will float in as it develops, because these cells here in this area here will develop into the embryo, and they will float in that amniotic fluid, and it helps to maintain a balanced temperature, and it comes from, in fact, the maternal blood, and later on in development of the embryo, it comes from the fetal urine. The other membrane that is developing is the yolk sac, and that comes from the hypoblast. Remember, the embryonic disc is made of epiblast and the epiblast and the hypoblast. Hypoblastic cells form the yolk sac, and it will go on eventually to form part of the digestive tube, and the very earliest blood cells appear in this area inside the yolk sac. And blood vessels, the very first blood vessels, it's the source of the most primitive germ cells or stem cells that will migrate to form gonads. So we covered the amnion and then the yolk sac, and the next one is, I believe, the chorion, no, the allantois. So here, the allantois. This is a sort of a outpocketing structure that juts out. It actually juts out to what's going to eventually be the tail end of the developing embryo, and it'll go on to form the umbilical cord. It also ends up being part of the urinary bladder, but let's stick with, that this area here, the allantois, will become the umbilical cord. And the last of the four extra embryonic membranes, or sometimes called embryonic membranes, is the chorion. And that's these cells in this area. Here, you see them here. And they help form the placenta. They're involved in placentation. And they'll enclose the embryonic body and all the other membranes eventually. So that's going to be the chorionic membrane. All right. Now I did want to say, I started earlier but I stopped, but I did want to say that there are a number of slides that have a great deal of detail, either in the diagram or actually in the text, that I don't want you to try to learn or memorize, and I'll make it clear which ones I'm talking about as I go along. Okay, so we're talking essentially about the process of gastrulation, as the blastocyst, which has embedded or implanted into the endometrium of the uterus, as it converts into a gastrula, which is during that process of implantation and also just after. So again, the inner cell mast or embryoblast will divide into an upper epiblast and a lower hypoblast. And this is what is called the embryonic disc. Now the embryonic disc will go on to form the three primary germ layers, the layers within the developing embryo that will go on to form all of the tissues of the body. And here we're getting into kind of more detail, certainly in the diagram. If you can make sense of this diagram, it's not easy because different views give quite a different appearance. Here is a kind of a top view down, and you're seeing right there, in that area there, what is called the primitive streak. And this here, in this diagram here, that's a side view of that, and you see just the part, a little part there of the primitive streak, and then it progresses on, and in this area here now you start seeing those three primary germ layers, the mesoderm, the ectoderm, and the endoderm. And that's these three layers that are forming. The ectoderm is in blue and the mesoderm is in reddish orange, I guess, or red, and the endoderm is in yellow here. So during this stage, you start to get these formation of these important structures and separation of different types of differentiation into separate different tissues that will go on to form quite different parts of the developing fetus. So that embryonic disc, remember, it's made of the epiblast and hypoblast. It undergoes cellular rearrangement, and you get what's called a primitive streak forming, and that actually establishes the longitudinal axis of the embryo. That's right down the middle of the embryo, and it transforms into the three-layered embryo. At this point, it's a three-layer. It goes to a three-layered embryo in here. All right, containing the three primary germ layers: the ectoderm, mesoderm, and endoderm. So all body organs, all tissues eventually will all derive from these three. All of this is the process of gastrulation, gastrula formation. The ectoderm, which is the layer that's right in contact with the amniotic cavity, will go on to form the nervous system and epithelial tissues, skin epithelium, also called the epidermis. The endoderm, which is in contact with the area called the yolk sac, the endoderm, will go on to form the mucous epithelial linings of the digestive, respiratory, and urogenital systems. So these are internal surfaces. And then the mesoderm forms everything else. So mesodermal cells will, which are present just below that primitive streak, will go on to form all the other tissues. And initially, you can find just below the primitive streak a structure called the notochord. So these, the endoderm and the ectoderm, are both considered to be epithelial tissues. So anything derived from them would be epithelial tissue. You have the skin epithelium and the epithelial lining of the internal digestive, what will be the internal digestive, respiratory, and urogenital systems that are forming surfaces with the outside. So gastrulation then proceeds to move on to organogenesis. And in organogenesis, you get formation of organs, organ systems. By the 8th week of development, all of them can be recognized within a 3-centimeter long embryo. So there's actually quite an amazing, huge explosion of cell growth and differentiation, enough that you can actually recognize structures and systems, organ systems. Very early on, the embryonic body I described before, which is differentiated into these three layers, ectoderm, mesoderm, and endoderm, during gastrulation, starts to fold. And you get folding along the length in that direction and on the opposite side. And then you also get a folding in this area here at the front end, or at the tail end and at the head. And the tail, at the front end, it'll start connecting with the yolk sac, and it'll be the primitive beginnings of the gut. This, of course, is endoderm. And up here at the top, the ectoderm will go on to form the notochord and the skin epithelium, and then in between is mesodermal derived tissues. Now this has more information than I would want you to know, but it covers what the ectoderm leads to, what the mesoderm tissue layer leads to, and what the endoderm leads to. And of course, this is all part of the developing epiblast, or this derives from the epiblast. And here you see in the diagram at the very top, you see the primitive streak and the three layers, the endoderm at the bottom, the ectoderm across the top in blue, and in the middle the mesoderm. Endoderm again will lead to epithelial linings of digestive, respiratory tracts and urogenital tract, also any glands associated with those. The ectoderm is the ecto meaning outside. Ectoderm leads to the epidermis, but also parts of the nervous system, the brain, the spinal cord, certain cells associated with that. And everything else comes from the mesoderm. Okay, there's way more information or structural changes described in these, and it can be very difficult to follow the progression, and the changes are occurring in a very defined sequence of events, and they are carefully timed, and they are occurring, really quite soon, one after the other. Here we see 4 images that are day 17, 2 days later 19, 1 day later 20, and then 2 days later 22. There are tremendous changes. Remember, underneath that notochord or neural, which will be the neural plate, underneath the primitive streak, you have the notochord, and this streak becomes the neural groove, and it curves around and joins until you get this structure, which will be eventually form the backbone, and at one end the brain. But the very first events in generation of organs or organogenesis will be the brain and spinal cord development, which I've sort of gone through. So the neural plate, the underneath the neural plate, you have the notochord, which goes on to form what's called a neural groove, and eventually a neural tube. And the anterior becomes the brain. The rest is the spinal cord. Okay, the remaining ectoderm, the part that doesn't become parts of the nervous system, will form the outer surface layer of the body, the epidermis, essentially the skin. And here you see the endoderm development of what structures the endoderm will lead to. It'll become the epithelial lining of the gastrointestinal tract and respiratory tract, which isn't written here, but that's what happens. Organs associated with the GI tract, also the stomach, liver, pancreas, gallbladder, all of these are associated and derived from the endodermal tissue. So it forms those epithelial linings of the organs and their digestive and respiratory tract. That's the endoderm. So we've discussed a little bit about the ectoderm and the endoderm, and we'll move on. This is the mesoderm, and here is where I'd say it's more detail than you would need. The notochord, which is just underneath that neural tube, or what used to be the neural plate that curled around and became a neural tube, the notochord is part of the mesoderm. and there are structures which I don't need you to know that will form and go on to form other parts, mesodermal derived parts of the body. So let's move on. I don't think this is necessary. It's too much detail. All of this is a bit too much, more than you really need. If you want, you have the information. You can refer to it if you ever need to. But let's move on. So the mesoderm specializes into all the other different tissues that I didn't mention when I was talking about the ectoderm, what tissues the ectoderm becomes and what tissues the endoderm becomes. So the mesoderm becomes all of these things here. None of it highlights properly. All of these, all of this, everything else. Quite a bit of tissue, huh? Okay, so let's talk about development of fetal circulation, which I may have, I don't know if I don't remember if I did or didn't talk about with you guys, but the fetal circulation, when it develops, is has some quite unique structures. And it starts off by the end of the third week, you have a pair of vessels, and where it's going to form into the heart, they fuse. But you have these two vessels at 3 weeks running up and down the developing embryo. And in one part, it becomes the heart, and the heart starts to actually beat. And you can see that very early on, autorhythmic beating. But there are some key developmental structures that are characteristic of the vasculature, the developing vasculature, which are only found prenatally and are only functional prenatally, and they are unique to the embryo and going on from the embryo to the fetus before birth. And they include the umbilical arteries, which are in the umbilical cord, and the umbilical veins, and 3 vascular shunts. A shunt is it's like a shortcut or a bypass. A bypass is a better synonym for a shunt. These shunts are absolutely required for normal development in the fetus. And the reason they are there, the reason they are needed or required, is because the fetus does not breathe air, does not breathe any gas, and does not eat. Okay, keep that in mind, please. It derives its respiratory gases by taking up oxygen from the maternal circulation, and any CO2 that its cells produce diffuse into its bloodstream and then diffuse across into the maternal circulation, and she breathes it off through her lungs. Also, any nutrients that the fetus needs are provided via the maternal circulation, diffusing nutrients across the placenta into the fetus. The 3 vascular shunts that are in the developing embryo and fetus, because of the unique characteristics of having the mother do, respiration essentially and providing nutrients. The 3 vascular shunts that are unique to the developing embryo and fetus are the ductus venosus, which bypasses the liver, the foramen ovale, which bypasses the pulmonary circuit of the vasculature, all the pulmonary circulation, and the ductus arteriosus, which transfers blood directly from the right ventricle to the aorta. And I'll describe them in more detail in the next few slides. So here we have a slide showing The fetal circulation, and this is quite advanced in terms of development fetus, and then in the newborn. Let's start actually at the bottom. This is the placenta, where the maternal circulation and the fetal circulation are in close proximity, but because the chorionic villi, they are separated by the chorionic villi but from each other, but they are in very close proximity, and gases diffuse across, oxygen from the mother to the fetus and CO2 the other way, and nutrients diffuse from the mother to the babe to the fetus, and metabolic wastes diffuse across from the fetus into the mother's circulation. The umbilical cord, which is comes from the placenta and then enters into the baby's, the fetus's abdominal cavity at what is now your belly button, so the navel. It has umbilical veins and umbilical arteries. Now, I remind you that the definition of a vein is a vascular structure that returns blood to the heart. So the umbilical vein in the developing fetus, which is bringing blood from the placenta up back into the fetus and back to the fetal heart. Okay, this is venous return. It has picked up oxygen inside the placenta. So this is fully oxygenated blood. All right, even though it is a vein, it's fully oxygenated blood. Now, normally, all the blood in the abdominal cavity, all the venous return, all the on the venous side of the vasculature, all blood first goes into, at least for organs in the peritoneal cavity, and these blood vessels are entering into the peritoneal cavity. All that blood would normally in an adult or newborn go first into the liver, because the returning blood also includes in a newborn and onwards into the adult blood that is draining from the intestines, and therefore it carries a lot of nutrients and maybe some toxins and has to be processed by the liver. But that's not the case in the fetus. There's no processing required. It's got whatever nutrient, all the nutrients are provided in a form that can be used, and there are no toxins, and there are no possible bacteria that might have to be cleared out that the liver normally does after birth and in adulthood. So that blood doesn't go into the liver. Remember, normally the blood would go into the portal hepatic, via the portal hepatic vein into the liver. It bypasses it because the fetus has a structure called the ductus venosus, and that's this structure here which takes the blood from the umbilical vein and provides a shunt or connection bypass directly into the inferior vena cava. Here it is, the inferior vena cava, this structure there. Okay, so that's the ductus venosus. Of course, I talked to you about the umbilical veins. It's bringing oxygenated blood back, bypasses the liver because of the ductus venosus. The blood goes into the inferior vena cava and then into the right atrium. Now, normally, blood, as you know, in a newborn or adult, blood will go into the right atrium, then to the right ventricle, and then up just like this, and then up the pulmonary trunk to the lungs to get oxygenated. But this blood has already received quite a bit of oxygen, and there's no air in the lungs in the developing embryo or fetus, so there's no point in sending it up there to the lungs, and therefore there are some bypass structures. One of them is a hole between the right atrium and the left atrium called the foramen ovale, which can shunt blood across from the right atrium to the left atrium. And the other structure is that the pulmonary trunk, is it labeled? No. Well, they're showing, anyhow, this is the pulmonary trunk right here. I'll label it. Here, this is the pulmonary trunk. Instead of going up to the lungs, what happens is that you have a structure unique to the embryo and fetus called the ductus arteriosus, which allows the blood to go directly across into the aorta. And then it spreads throughout the body, and eventually via this abdominal aorta goes down through the, will go, oops, that blood will eventually lead down and go to the umbilical arteries and down the umbilicus to be fully oxygenated here in the placenta. Well, after birth, all of this detail is written on the next few slides. After birth, these structures close, although there is some delay in closure of the foramen ovale. It becomes, I think I have it in the slides, it becomes fully closed off by a year. It still remains slightly open, but then it'll close off. I believe that's correct. The others almost immediately at birth close off. And here that's described again, some of it maybe in more detail or in a different way. I took this slide as with some, a repeat of the information, some other information, but maybe a better, more easily understood in some ways. Diagram from your book. There's the foramen ovale going from the right atrium across. Here's the ductus arteriosus. It's actually sometimes even a bigger structure curving across from the pulmonary trunk into the aortic arch. Here's the ductus venosus bypassing the liver, not sending the blood into the liver via the portal hepatic vein, but going past it into the inferior vena cava. Okay, and this is a pathway diagram showing the pathway of blood, and it actually shows what would normally happen, and a little bit of blood does go through where it normally does, but most of it is traveling through this modified circulation in the fetus. So there's a comparison here between the fetal circulation and the newborn. What you have is starting at the placenta in a fetus. Now I'm talking about the fetus. It goes to the umbilical vein and then bypasses the liver via the ductus venosus, which joins the inferior vena cava, and then that feeds into the right atrium. And some of that blood, a good amount of that blood, actually goes straight across through the foramen ovale into the left atrium, then the left ventricle, and gets pumped out through the aorta. But also some of the blood goes from the right atrium to the right ventricle and into the pulmonary trunk. But then instead of going to the lungs, it goes through the ductus arteriosus that joins up with the aorta. And part of the blood that's pumped out of the aorta, a good part of the blood that's pumped out of the aorta, will work its way through the umbilical arteries and go back down through to be reoxygenated and dump CO2 at the level of the placenta. Normally, once birth occurs, this ductus venosus, let's do it in a different color maybe, this shunt, so the ductus venosus is not present, nor of course are the umbilical veins and blood, all the blood draining from the cavity of the abdomen will go into the liver. I think what I'll do is, well, some of that blood, a very small amount of that blood, of course, actually does go. Let's do a different, very small amount of the blood. All of, most of it goes from the umbilical vein to the ductus venosus, etc. A very small amount of it will go into the liver because the liver needs some oxygen also. So it does a little bit go through there, and a little bit of the blood from the pulmonary tongue doesn't go into the ductus arteriosus, but actually does go through the lungs and then back to the left atrium, because again, that tissue does need some oxygen, but it doesn't take much. So this blood is providing oxygen to the tissues. And that's the end of Unit 8B, Part 1.